Chapter 13 deals with the Krebs cycle, also called the citric acid cycle, as well as oxidative phosphorylation. Let me just say a few introductory comments about the citric acid cycle. Much like glycolysis, which most medical students don't appreciate or don't like for a pathway, citric acid cycle is yet another pathway which students don't like or don't care for. But let me just say a very important issue about the Krebs cycle. Even though students don't like it, it is the most important pathway for life. You and I cannot live without the Krebs cycle, the citric acid cycle. Even glycolysis, some people live with a partial enzyme defect, not a complete enzyme defect, a partial enzyme defect. You saw that already with pyruvate kinase deficiency. But the Krebs cycle, in your lifetime, you will never see a patient who has even a partial enzyme defect. And the reason is it is essential for life. This pathway, the citric acid cycle, is the most important pathway for life. And so at least give it some respect, even though you may not like this particular pathway. Now, why is this pathway so important for life? And in a nutshell, it's right here. And the reason is because the acetyl-CoA, this comes from the PDH enzyme, which comes from glucose as well as fatty acids and amino acids. There's 2 carbons of the acetyl-CoA. They go into the Krebs cycle, and they see a number of enzymes that we'll be talking about, and completely oxidizing aerobically both carbons of the acetyl-CoA to CO2 and water, generating 12 ATP equivalents. And so each glucose generates 2 acetyl-CoAs. Each glucose generates 24 ATPs. But each acetyl-CoA generates 12 ATPs times 2. That's where the 24 is coming from. This is why this pathway is so important for life. Because without this pathway, you cannot get enough ATPs to carry out all those membrane potentials in the brain, all those transporters in the kidney, all those muscle contraction that has to go on. Most of the ATPs is being generated for those procedures by this particular pathway. Yes, you get a little bit in glycolysis and other pathways, amino acid metabolism, but most of the ATPs come here. If it's a defect in the pathway, incompatible with life. Okay, so let's talk about this pathway, the Krebs cycle, also called the tricarboxylic acid cycle, the TCA cycle, the citric acid cycle, whatever you want to call it, as long as you understand the basic principles of this particular pathway for life. Now, one comment to make about this pathway is, unlike other kind of pathways like glycolysis and other ones we'll be talking about, this pathway, there's no hormonal control. There is regulation of the pathway, but no hormone like insulin or glucagon or epinephrine regulates it directly. So when you and I eat glucose or eat amino acids or whatever, we'll talk about the amino acids in a later chapter. What happens there is in the pathway of glycolysis with the glucose, the end product is pyruvate. Some amino acids also can generate that pyruvate, but then the pyruvate leaves the cytoplasm, gets into the mitochondria, and sees the enzyme PDH. That was at the end of chapter 12. Now, PDH, that pyruvate dehydrogenase, what the enzyme is going to do is take the 3 carbons of pyruvate, oxidizes it and removes the electrons, puts them on the NAD to form NADH, and then to get 3 ATPs there. Because it decarboxylates, you blow off CO2, and then generating acetyl-CoA of a product. Now, the 2 carbons of the acetyl-CoA then sees a low-yield enzyme, condenses the 2 carbons of the acetate with the 4-carbon oxaloacetate to make the 6-carbon citrate. Now, before we move on, let me just mention, in this cycle, there are a number of enzymes. In my opinion, just 2 enzymes you need to know from the National Board. All the other enzymes, in my opinion, are low-yield enzymes for the boards. Even this enzyme, citrate synthase, is a low-yield enzyme. I realize other textbooks recommend you know this enzyme. I've never heard about this enzyme being on a national board. It is, in my opinion, a low-yield enzyme, the name of the enzyme that synthesizes citrate. Now, other 2 enzymes that are important to know, there'll be one very major enzyme and one subservient enzyme. I'll cover those enzymes in a little bit. Okay, so we get condensation of the 2 carbon of the acetyl-CoA to the 4-carbon OAA to make the 6-carbon citrate. Let's proceed. The 6-carbon citrate sees a low-yield enzyme, don't worry about it even though it's listed here, to form isocitrate as a product. Once isocitrate is generated, it sees an enzyme which, notice the surname, dehydrogenase. So again, when you see that enzyme, dehydrogenase, that means oxidation reduction. And what this enzyme is going to do, it's going to remove high-energy electrons from isocitrate. It puts them on the NAD to form NADH as a product. Now what happens to the NADH? It goes in the electron transport chain right there in the mitochondria. And then the electrons ultimately will go on to the oxygen. That's why this pathway is oxygen dependent, because when the electrons coming from NADH have to go somewhere, they're going on to the oxygen. So those electrons go into the electron transport chain, generating ATP. The electron transport chain will be discussed later in this particular chapter. Now, what else does this enzyme do? What this enzyme does, besides carrying out oxidation, it also decarboxylates, and one of the carbons is now blown off as carbon dioxide. And because one of the 6 carbons is blown off, that gives rise to the 5-carbon product, alpha-ketoglutarate. Let's proceed. Once alpha-ketoglutarate is generated, sometimes abbreviated alpha-KG, it sees an enzyme called alpha-ketoglutarate dehydrogenase. Now notice the name of that enzyme. The surname is dehydrogenase, meaning it's going to carry out oxidation reduction, pluck out a couple more electrons from alpha-ketoglutarate, puts them on the NAD to form NADH, and then the NADH will give electrons ultimately to the oxygen that you breathe in, generating even more ATP. Now, this enzyme also decarboxylates, generating CO2 as a product. So notice here the 2 carbons coming into the cycle from acetyl-CoA and 2 carbons being blown off as CO2. So both carbons come in and both ultimately are blown off as CO2. And then because a carbon is blown off as CO2, we now end up with a 4-carbon product, succinyl-CoA. So we went from the 6-carbon isocitrate to the 5-carbon alpha-ketoglutarate to the 4-carbon succinyl-CoA. Now, why is this enzyme highlighted in red? This enzyme, again, just 2 enzymes in the pathway you need to know from the National Board, a subservient one, and this is the subservient enzyme. The other one is more important. But why is this enzyme highlighted? Why is it highlighted? There's no pathology associated with it. There's no pharmacology associated with it. The reason why you need to know it is what it needs to work to carry out this one arrow in biochemistry. And a clue as to what this enzyme needs to work is, notice that this enzyme generates NADH, splits off CO2, and makes a coenzyme A of a product. There was yet another enzyme that we talked about in either Chapter 12 or Chapter 13, which we're at right now, that generates a coenzyme A of a product. And that enzyme uses the same coenzymes that carry out a similar step. This enzyme, as well as the other enzyme, requires tender, loving care for Nancy as a product. T stands for thymine pyrophosphate, L for lipoic acid, C for coenzyme A, F for FAD, and N for NAD. Now, this is the second of 3 enzymes in your body that is a tender, loving care for Nancy enzyme. And what was the first one that was discussed? And the answer is right at the top here. That's PDH. PDH generates NADH, splits off CO2, and makes a coenzyme A of a product, acetyl-CoA. So this is the second. This one down here, alpha-ketoglutarate dehydrogenase, it's the second of 3 critical enzymes in the body that requires tender, loving care for Nancy as coenzymes. That's why it's so important to know. And before we get to that in a later chapter, can you think of the 3rd enzyme that requires tender, loving care for Nancy as coenzymes? And in fact, that 3rd one may be even more important than these 2 here. And the reason is there is disease associated with the 3rd one. And that disease is maple syrup urine disease. But that will be discussed in a later chapter. Let's proceed. So it makes succinyl-CoA as a product. Once succinyl-CoA is generated, it sees a low-yield enzyme. Don't worry about it. But what this enzyme is going to do, it's going to take the high energy of succinyl-CoA. A coenzyme A of a product is a high-energy molecule. What this enzyme is going to do is take the energy of succinyl-CoA and captures it and couples inorganic phosphate and GDP to make GTP, which is equivalent to ATP in terms of energy. Because an ATP is being generated, an ATP equivalent is being generated, you can consider this arrow, even though the name of the enzyme is not important, you can consider this arrow as the third and last example in your body of what's known as substrate-level phosphorylation, meaning a substrate was used to phosphorylate, in this case here, GDP to make GTP, or phosphorylate ADP to make ATP. A substrate was used to make an ATP equivalent, the third and last example. Now, I know what you might be thinking. What were the first two examples that were discussed? And the first two examples were in glycolysis, where a substrate was used to make an ATP equivalent. One of those examples was 1,3-bisphosphoglycerate, was used to make ATP directly. And the second example was phosphoenolpyruvate, PEP, which was used to make ATP directly. And this is the third example, is succinyl-CoA. Just in case the National Board asks you which one of the following molecules can be directly used to make an ATP equivalent. So you need to be ready of those three kinds of substrates that can be used to make ATP. I want to stress that substrate-level phosphorylation is different than oxidative phosphorylation, where you make ATP from another way, and that's this thing called the electron transport chain. The electron transport chain can be used to make ATP. But here, a substrate is directly used to make ATP. Okay, so anyway, let's proceed. We generate succinate as a product, the 4-carbon succinate as a product. Then once succinate is generated, it sees a low-yield enzyme. Don't worry about it. I won't even name the name of the enzyme. But what is important about this enzyme, that this enzyme is located in two pathways. One pathway here called the Krebs cycle, and the other pathway that it's located is coming up in the later figure. It's in the electron transport chain, and there that enzyme, even though you don't need to know the name of the enzyme, generates FADH2 because it takes electrons from the succinate and puts them on the FAD to form FADH2, which goes in electron transport chain, generating 2 ATPs from the FADH2. Now, in the other pathway, that enzyme, even though you don't have to know it, it's called complex 2. So in this pathway is a name called succinate dehydrogenase, low yield. But in the other pathway called electron transport chain, that same enzyme is called complex 2, that you'll see later on in this chapter. Now, the other product of the end of this arrow is fumarate, as well as the FADH2. Now, the fumarate, once it's formed, low yield, it sees an enzyme, low yield, generates malate. Once malate is formed, it sees yet another low yield enzyme, but it is a dehydrogenase, which means oxidation reduction, meaning electrons, high energy electrons, are being plucked out of malate, put on the NAD to form NADH. NADH then will give the electrons ultimately to the oxygen in the electron transport chain, generating even more ATP. Now, the product of this enzyme besides NADH is oxaloacetate to reinitiate the cycle. So to summarize this cycle, is that the cycle is, what it has is a number of enzymes that are efficiently plucking out these high energy electrons of the 2 carbons of pyruvate, blowing off both carbons as CO2 and water. And its end product of all that metabolism is the carbon dioxide. That's why you breathe out all that CO2 every few seconds, the end product of this particular pathway primarily. Now, the other product, the other idea here is it's making ATP equivalents in this pathway. Now, how many ATPs are being generated in the Krebs cycle? For the National Board, nowadays, number crunching and how many ATPs you get here versus there, that is much lower yield compared to the previous years of the USMLEs. Nowadays, when you're down to about 336 questions for the boards, number crunching is much lower yield. But just in case they ask, just to cover the bases here, how many ATPs you get in the Krebs cycle, the answer right there is 12. Where are those 12 ATP equivalents coming from? 3 from the NADH here, 3 from the NADH here. Here's the GTP, 1. Here's 2 from the FADH2, and 3 from the NADH. So one more time. 3, 3, 1, 2, 3. So 12 ATP equivalents per cycle. That's a lot of ATP, and that ATP, again, will be used for muscle contraction, for brain function, for kidney function, anything that's going on in the body. ATPs, most of it comes from this particular cycle. Okay, now, we're not done with the cycle yet because we got to talk about the regulation of the cycle. It's not under hormonal control, but it's highly, highly regulated. And so what regulates this pathway? Well, what regulates this pathway, we now have to talk about the most important enzyme of the pathway. So which one of these enzymes is the most important enzyme of the pathway? Let me highlight it, and that enzyme is isocitrate dehydrogenase, the most important enzyme of the pathway. Again, why do you need to know that enzyme? There's no path or farm associated with it. It's highly regulated. And what regulates this enzyme is high levels of ATP and high levels of NADH. So now, do high levels of ATP and NADH turn on the enzyme or turn it off? I can give you the answer right away, but if you think about it first, it makes sense. Now think about it. If you were to eat a high carbohydrate meal, glucose enters all the cells in your body, runs through glycolysis, getting a few ATP there, goes into mitochondria, pyruvate goes into mitochondria, generating 3 ATPs by PDH enzyme, and then the acetyl-CoA product goes into the Krebs cycle, generating 12 ATPs. So as you might imagine, that when you eat a high carbohydrate meal, the Krebs cycle is now working and working and working, cranking out the ATPs, the NADH, the ATPs, the NADH. Eventually the cells say, hey, enough is enough. We don't need any more ATP. Shut down the pathway. And sure enough, it shuts down right there with the isocitrate dehydrogenase. This enzyme shuts down by high levels of ATP and high levels of NADH because we don't need any more if they're not being consumed. So now notice what's going to happen when the enzyme shuts down. Then isocitrate, the substrate, starts to accumulate. It backs up to the citrate in the mitochondria. Then citrate in the mitochondria will start to accumulate. It spills out of the mitochondria by a transport system, and citrate now enters the cytoplasm. And when citrate enters the cytoplasm, does that ring a bell? Hopefully it does, because when citrate enters the cytoplasm, it sees the top gun of glycolysis. And what was the top gun of glycolysis? And that's the enzyme PFK1. Now, in the earlier chapter, in chapter 12, I had mentioned that PFK1 is inhibited by ATP, which makes sense. If you have a lot of ATP around, why make more ATP? Shut down the glycolysis. And also inhibits PFK1 is citrate. And at the time I mentioned in chapter 12 that it may not make sense yet, but hopefully now it makes sense. Why is citrate chosen to shut down PFK1? And the reason is because when the Krebs cycle shuts down, the citrate gets out in the cytoplasm of all the cells, and says the glycolysis in the cytoplasm of all the cells in the body also shut down by inhibiting PFK1. But you got to remember, in the liver and the liver only, there was something special going on. Where in the liver, remember, when you eat this high carbohydrate meal, why is glycolysis working, whereas in all the other cells shut down? Because in the liver, insulin turns on PFK2, which makes fructose 2,6-bisphosphate, which overrides the ATP citrate inhibition. So in glycolysis in the liver, glycolysis is working overtime, whereas in all the other cells in the body, glycolysis comes to a grinding halt because of that insulin activation of PFK2. So again, now the issue is in the bigger picture of life. Again, why is glycolysis working in the liver, whereas in the other cells it shuts down? Because here it is, when you eat this high carbohydrate meal with all that glucose, you run glycolysis, you run PDH, all the way down to the citrate, and then all the ATP will shut down isocitrate dehydrogenase, but now the citrate backs up, gets out in the cytoplasm, and as we'll talk about later on, out in the cytoplasm, there is a pathway called fatty acid synthesis. So in a later chapter, we'll talk about how you take sugar and convert that carbon of sugar to the carbon of fatty acids. And again, it involves glycolysis and the Krebs cycle to get the carbon out of the cell into the cytoplasm to carry out this very important pathway called fatty acid synthesis, which is going on in abundance in the liver. I cannot emphasize enough, this is going primarily in the liver, because other cells in your body, glycolysis shuts down because the PFK1 cannot be overridden. Only in the liver is it overridden. Okay, so this is the Krebs cycle and why it's so important.